Cells undergo a lot of changes during mitosis, which must all be coordinated to occur at the right time and place. The cytoskeleton reorganizes into the mitotic spindle, while the centrosomes separate to form opposing spindle poles. The Golgi disassembles, chromosomes condense, and the nuclear envelope breaks down. Central to these events is the cyclin-dependent kinase CDK1 and its regulator CyclinB, proteins that have long interested Jonathon Pines from the Gurdon Institute in Cambridge in the United Kingdom.
CyclinB1-Cdk is both the trigger for mitosis and, as Eric Nigg once put it, it is also the workhorse for mitosis. So it does a lot of the phosphorylation of substrates that cause the complete reorganization of the cell architecture in mitosis. When I was a postdoc, I found that it had this interesting localization change at mitosis. It started in the cytoplasm, and then all of a sudden most of it ended up in the nucleus. After I set up my own lab, we showed that it very, very quickly goes into the nucleus at mitosis. Just before you get all the changes in the nucleus that you can see down the microscope. One of the key questions was exactly what triggers CyclinB to move at just that point in mitosis.
A postdoc in Pines' lab, Olivier Gavet, developed a probe to measure CyclinB1-Cdk1 activity in living cells. When the probe is phosphorylated by the kinase, it undergoes a conformational change that increases the Forster resonance energy transfer between the probe's cyan and yellow fluorescent proteins. By comparing the kinase's activity to its localization, Gavet and Pines found that CyclinB1-Cdk1 translocated into the nucleus as soon as it became active in early prophase, and that continuous kinase activity was required to maintain it there before nuclear envelope breakdown. Adding a CDK inhibitor caused CyclinB's rapid re-export from the nucleus.
That immediately suggested a quite simple but rather elegant way in which you could synchronize the changes in cell architecture in the cytoplasm and the nucleus, that when you turn it on, CyclinB begins changing the cytoplasm, but if at the same time you trigger a proportionate to move into the nucleus, then you would have automatically the nuclear and cytoplasmic changes being synchronized.
Previous research suggested that the mitotic kinase PLK1 boosted CyclinB1-Cdk1's nuclear accumulation by phosphorylating CyclinB to prevent its export from the nucleus. But inhibiting PLK1 had no effect on CyclinB's nuclear localization. Nor did CyclinB accumulate in the nucleus because of a block in its nuclear export.
If we use a nuclear export inhibitor such as leptomycin B, we find that makes absolutely no difference to the nuclear import rate of CyclinB. So the import is not down to just turning off export. What it's down to is when CyclinB1-Cdk1 activity turns on, it causes a 40-fold increase in its nuclear import rate.
Previous studies from Pines' lab indicated that CyclinB1-Cdk1 could stimulate its own nuclear entry by phosphorylating itself to create a nuclear import signal.
Previously, we found that if we made mutations in some conserved CDK phosphorylation sites in the amino terminus of CyclinB, you could change its import. What we found now is that you do indeed change its import, but it really only makes a difference in interphase. When you get to mitosis, this vast increase in import rate, it doesn't matter if you have the phosphorylation sites there or not.
So CyclinB1-Cdk1 triggers its own import, but doesn't do so by phosphorylating itself. Gavet and Pines turn their attention to the nuclear import machinery instead.
We haven't yet managed to get to the bottom of this, but we did make this rather intriguing observation that at the same time as you get this 40-fold increase in CyclinB1 imports, you also begin to get export of a substrate of the transportin nuclear import pathway called N9. So this increase in import rate seems to reflect a general change in the nuclear transport machinery.
There's some specificity to this change, because other proteins don't alter their localization until well after CyclinB's nuclear import. The CDK activator, CDC25 for example, only enters the nucleus as the nuclear envelope starts to break down. So how does Pines think CyclinB1-Cdk1 influences the transport machinery to induce its own rapid accumulation in the nucleus.
Potentially, either changes in the nuclear import carriers themselves, or it could be that you're getting a change in the nuclear pores. There's now a number of studies that you have quite dramatic changes in phosphorylation of the nuclear porins, and obviously that could change affinities for particular substrates, or change the permeability of the pore because the pore itself begins to disassemble. A number of the phosphorylation sites on the nuclear porins are themselves CDK sites. So our favoured hypothesis at the moment would be as you turn on CyclinB1-Cdk1, it begins to phosphorylate components of the nuclear pores, and that creates a change in the permeability of the pore, which then increases the import of cyclin B and the export of this M9 substrate.
However CyclinB1-Cdk1 stimulates its own import, the fact that it does so without the help of other kinases like PLK1 simplifies the current model of mitosis and its coordination in the nucleus and cytoplasm.
We don't have to have many different things coordinated for triggering mitosis. We shift the focus back to the main step, the critical step, being the activation of cyclin B itself. So we're pretty sure that cyclin B starts to be activated in the cytoplasm, and then the question is where in the cytoplasm. A few years ago we postulated that maybe it starts to be activated on centrosomes. We've tried to put the probe onto centrosomes, but unfortunately everything we've done so far to target it has killed the threat. But we have other ways that we're trying to address that. It might be rather exciting to see a wave of activity beginning to spread from particular nucleating centers in the cytoplasm. That would be very interesting to see.
You can read more about how CyclinB1-Cdk1 stimulates its own nuclear import to synchronize mitotic reorganization in the paper by Gavet and Pines in the April 19th edition of the Journal of Cell Biology.